My job is to make sure that nobody falls asleep. So,
Tough job.
our next keynote speaker is Dr. Chen, University of Alabama, Birmingham. Morning we're talking about the multidisciplinary approach of bioinformatics. I think the best example is undergraduate in basic sciences and then computer science degree later. And he's a pioneer in translational bioinformatics, data mining. So he's going to talk about unraveling the complex patterns in omic data with integrated GNPA tools.
All right, good afternoon. Thank you very much for the nice introduction and coming back. I'm a bioinformatician, so I'm going to actually talk to you a little bit about my experience developing bioinformatics tools to solve some problems in translational bioinformatics. Since we're in the meeting context, so I can't resist myself advertising a related meeting. So we're in the Mid-South regions that include Louisiana, Oklahoma, Alabama, and so on. There's an organization called the Mid-South Bioinformatics Society, and they are having annual meetings, and this year just concluded in Mississippi. Next year, we're going to host it at Birmingham on March 28th through the 30th, and the theme is informatics for precision medicine. So if you felt that this is a good topic, and please do come to this related conference as well. Today I'm going to actually break up my talk into the three sessions. And the first is really talk about this big data and small data and the need for models. And then the following two sections will talk about our research in this integrated gene ontology network and pathway analysis tools. So I think yesterday one of our keynote speakers already used this slide. I think what this slide shows that the NCBI keep track of the data increase, both in terms of users and the size. That is actually, it's not YouTube 2015, it's 2025. So essentially, NCBI is providing a YouTube for the cells, and so we're addicted to the content, and the content is going to grow very, very significantly. But as a bioinformatician, I don't like the fact that we're dealing with big data. Maybe as an IT person, I would like it. I really like this other slide that NCBI provided. They're trying to actually reduce the data. Let's say this, initially the 1,000 Genome Project produced 250 terabytes of data. And by using just compression techniques and then coming up with a very short list of the significant variants, and then essentially you come up with small data. And I think it's the small data that, as a bioinformatician, I prize myself in, because that really distills the information. As human beings, we may not be able to deal with all of these data in our whole life. So I think there is a theme how to deal with this data. So starting from a phenotypic state, maybe health, disease, and then we'll do a lot of molecular measurements. And this process from right to left is what I consider digital scanning. And that's where the big data is getting produced. And we're actually analyzing this information. I think it's essentially mapping this data back to the phenotypic state. And in this process, it's imperative that we make this filtering, ranking, inferencing to happen such that we get abbreviated knowledge. But the bumpy road is really bumpy. And here's a picture about the promise of precision medicine. So imagine that we did all of the NGS analysis, we figure out what just a cancer can be actually related to what target mutation. Right? So in this particular scenario, and then we use a drug A. And then if we find the same cancer organ, a different mutation, we use a different drug. And if there's the same mutation in the different parts of the body, then we use recycle the same drug. So it seems a very simple paradigm, and in fact, the NIH have conducted trials. But 2 years ago in Nature, there's an editorial that talked about the disappointing outcome of precision oncology. According to all the trials conducted at Texas MD Anderson and the NCI, so the attempt to pair the mutations with the target with the drugs has only produced a very marginal increase in the improvement in health outcome. That's only about 2 to 5%. So the rest of it is either we don't have the right drug, or the cancer changes, or we would have treated this patient in the same way originally without the knowledge. So I think it turned out that this is non-trivial. I think if you think about it, the reason it's non-trivial is maybe the way that when we reduce the big data to small data is biased, or maybe the biology is so complex. In the case of cancer, you know that cancer is not just a messy cell waiting to be killed, and it's a whole process that starts from a genome instability, and there's content frequently released through this just a palette, and then it just interfere with the cancer metastasis and create a microenvironment that really feeds itself. So this process is really dynamic, and so how do you really use bioinformatics to study this? And we can't really treat it as a constant, stable state thing. So now I come back to this integrative analysis. So usually when we try to do bioinformatics, we start with the big data that usually is capturing the complex system as specific conditions and time point. And then we do a lot of the so-called upstream analysis, and that's reducing the data through a pipeline down to some statistical, really significant list, right? And then I think this part is, I'll talk about more details later. It's an integrative omics analysis, and then you throw all kinds of tools try to make sense of this data. But sometimes the data we generate as bioinformaticians, when we're not careful, we generate more pathway diagram than we started with. So have to be very careful. And then the downstream analysis where biologists start to do wet lab work. So it's this part that is very heterogeneous, and there's no community standard. And I think the questions here is, can we characterize these large omics data set in their complex molecular system context so that we can achieve some really discovery insight? I think that these are all the things that most of the bioinformaticians here are familiar with. So you do gene set analysis, pathway analysis, network analysis, and then the gene set analysis help you figure out what gene ontology category is enriched in the data set. Pathway analysis help you map the genes that's altered in the pathway context. Network analysis and help you prioritize genes. But then they all seem to be just independent of each other. Right? So there's an inspirational case study we did that's more than 10 years ago. Here's a network, but this network has nodes and edges. The nodes are not genes. The nodes are actually, in this particular study, are enriched functional categories that represent collections of genes from a particular study. In this particular study is an ovarian cancer drug resistance. We did the proteomics analysis that compare the drug sensitive and drug resistant cell lines, and then we harvest the list of the proteomics data set, and then we did the gene ontology. But instead of showing the gene ontology category in the list, and we show them as nodes. And then the edges are actually what we call the crosstalk between the gene ontology categories. And the red edges are actually enriched crosstalk. And how do we do the crosstalk? And there's a paper that talks about the detail, but essentially we look at the enriched protein interactions between two functional categories. So why is this inspirational? Because then all of a sudden the complexity of the network gets reduced significantly. You don't have to look at the sticky hairball-like structure and try to make sense of the data. And in fact, we use this and we show, oh, this is mostly endogenous response to stress, and less about the endogenous response to cell stress. So we pretty much rule out the hypothesis in ovarian cancer drug resistance, where people previously thought that the resistance would develop because cell has less transporters on their surface, and therefore mechanically resisting the drug. It's really induced a broad spectrum of gene expression intracellularly. Okay, so I think then we're pretty much convinced that that some kind of a model like that is useful, but I think that there's a need for creation biosystems models. And so when you think about if you're just doing it without a model, this is pretty common. You treat this as a cell line or samples, and you do all kinds of perturbation. You get the big data, and then you try to use this omics data to try to infer what's happening. But if you're trying to use a model representation, you try to understand what's inside of this cell that can be mapped to different nodes and connections of nodes. Ideally, you should be able to do it quantitatively using differential equations, but we never can because the cell is not in a static state, and there are always different kinds of compartments. You can't capture all the parameters of the reactions, but I think we're working toward that. Maybe at least have some kind of structure that can allow us to inference what's going on with given input. So I think that there is a systems biology component. I want to go through the details. So essentially, what I'm actually advertising is for any kind of tools that we develop as a power bioinformatician, we need to add an extra step. And this extra step is, instead of just directly shoving the data through filtering statistics, we need to actually build dynamic systems models through this modeling. And then we use these models to help guide us. And the rest of the talk, I'm trying to convince you that there's a virtue in doing so. I think in terms of the representation of this model, biological networks has been around for many years, and I still think that the network model has virtue because you can both use it to study the biology connections, and some connections, the loss or gain of it can cause disease. And you can also use this as essentially a data representation to study how the disease evolve over time or over different conditions, and so that you can do dynamic modeling. But what I also want to point out is that the network is not just about the connections and the components. There's also this community concept, just like what we learned in the social network. Right? So this is a paper years ago by Biosystems group that shows that there are different communities that we can discover. If you're using topological network, let's say the edges represent the protein physical interactions among nodes that are represented as genes or proteins, then there are actually tightly connected clusters. And in this community, you're more likely to find protein complexes. if you use functional module and then the edges represent the co-expressions, and then you're more likely to get the functional gene expression clusters. Or if you're using curations of diseases, and then you get different communities. So this community concept is also very important. Well, now the question is, can we map them together? So I think our way of mapping them is to use a data structure that called a PAG. And PAG represent pathways, annotated lists, or gene signatures. You say, well, these are all biologically different concepts, but as a computer scientist, we like to simplify things a little bit before we can get complex. So pathways are, in my opinion, so they are just collections of genes with some structures, and these structures are previously well documented, supported by literature. And annotated lists are things that you collected from literature or DD-GAP database, and you suspect that they're connected, but you don't really know anything other than the list. The gene signatures are more of a gene set that you collected from Omics data set. You can't explain them, but you know that they're real in a specific context. So I think that's the very basic data structure. In fact, we had a standard representation and database for it. And we use these 3-letter code to indicate where it's coming from and whether it's computational, experimentally derived, or any kind of details that's been captured. And then we just capture the information about genes within the PAG and additional details. So we have a paper. You can look at the details how we standardize the naming, but by giving them standardized naming conventions, apply those to these PAGs, we can now just analyze all of them together. And so in terms of the data sources, you can see that we are essentially going all across the board, databases from NCBI, EBI, or even some of the lab. For example, the annotated list that would include a GAD database, GWAS catalog, or even EWAS studies from clinical database from Unravelling. And then expression signature also involve the drug perturbation or drug-induced gene expression signatures. And even microRNA target, we put them into annotated list. So it's very comprehensive, not just gene ontology or pathways. But there's one issue. So if you put them all together, how do you know that the quality are good, right? So we don't want someone to randomly come up with a list and throw it into the database. So we come up with a score that essentially based on a very simple statistical test. And this score, we call it COCO. So it's two-part, CO-I and CO-T. And CO-I is to look at, let's say this is a PAG. We're looking at all of the interactions that's captured among all the genes inside a PAG versus all of the interactions that can captured outside. And then we're comparing all the theoretical possible interactions inside a PAG and all the theoretical possible interactions outside. We use these 4 parameters and then plug in the same hypergeometric distribution to generate a p-value. And then the COCO-I score is by counting interactions. And CO-T is by looking at the triangles, enrichment of triangles. So in this particular case, inside is 3 and outside is 1, 2, 3, 4, 5. So, so we look at these information and we actually were able to show that when you're using CoI, and when you're looking at the randomly generated path, and the score is roughly at this level. And when you're looking at the content from our database, and that's very significant. Also, CoT is very significant. And then why do we put the two of them together? Is primarily when we look at the precision recall performance of retrieving true path by giving a varying degree of threshold based on the cutoff threshold, and try to see whether the true paths were retrieved. And we found that either CoI or CoT, they have different performance at the different size, and CoT may be slightly better when the size is small, and the CoI is better when the size is large. But when we put them together, the performance is better than each of the individual ones. So, and then we also did some work on looking at the size bias and corrections. This is another paper we published earlier this year at NAR. So you look at the pack size, and you look at the possible scores. And there seems to be a bias, and the larger the pack size, the bigger the score. We know that in cancer pathway, there could be 3,000 genes in the wiki pathways just on cancer. And then there are some of the pack that are so small, just 3 or 4. So how do you adjust for the potential size variation? And then we talk about a normalization method. And sometimes the normalization may be useful if you want to compare the information content across different variety of packs that's retrieved. And that may not be a problem that you see when you're comparing pathways of similar sizes, but when you're comparing things of such a variety, it seems to have come out. So in this particular example, this is a pad. Initially, it has a COCO score smaller, but after adjustment for this small pack, it's very biological significant. And then this other one initially is large, and initially was large, and then after adjustment, it comes down. Okay. I'll skip this and essentially continue to say that the information is very significant even after we adjust for the size. In this database, we also look at the so-called relationship among paths. If we don't look at the relationship, it's just yet another pathway and gene ontology integrated analysis. So we define the relationship as several types, two major types. One is this kind of relationship. We have two paths, A and B, and they overlap some genes in their intersection. Okay. It's very common, and if you have done just standard pathway analysis, you'll see that ER signaling pathways and cancer pathways and P53 pathways, they all have overlapping component. And sometimes that those standard analysis doesn't tell you how to reduce it further. So I think that we use this representation to indicate without any arrows to represent that they have a relationship, and then we use a simple hypergeometric distribution test to find out such relationship pairs. All right, and I won't bother to show you the details, but essentially this is a result of constructing a network for a colorectal cancer proteomics case study, and this is also published. So each of the node here represent a potential path. And this color indicate where the path may come from. Some come from tag, like all the gray ones. Some are from Biocolor. It's another pathway database. Some come from 6ALCCBB. That's the gene signatures down here. So these lines and the thickness of the lines to indicate are used to indicate how significantly they're related to each other by this shared membership relationship. So you can see that part of it is connected. And you may wonder, why can't we just integrate everything into one huge diagram? I think that this may not be scientifically valid. If you talk to any two scientists, and they would argue that each one of them may act on different signaling input and work independently. So that's the best way that we can do to consolidate without forcing everyone to conform to one view of science. So how do we use this? So we map the cancer data overexpression here, and you can directly see, even just visually, that most of the changes are in this part of the regions, and some of the changes, because they're not highlighted, they're not enriched per se. So all of a sudden, you're looking at one figure. You can still go into the individual pathways, but I think you would probably side with me that this is not as uglier as before. And then we also can construct a different second type of relationship called a regulatory relationship. And that's a relationship that essentially says, is A upstream of B? So that's a very practical question to ask. If you think about a single molecule, so is A transcription factor of B gene target? But I think in the context of PAGs, it's also important to ask those questions. So again, we come up with simple statistical tests to look for enrichment of regulations. It could be transcriptional regulations from transfact database, going from A to B. And then we found that in the database, the co-membership based versus the co-regulation based just PAGs with different degree of centrality, they're not necessarily always linear. The trend is there, but there are certain PAGs that tend to show up in more in one type than the other. So for example, especially the cell cycles seem to be heavily used in the regulatory PAG network, which is not surprising because cancer is well studied and a lot of the genes are either upstream regulating cell cycle or downstream. And so there's my argument is that there's a utility in not only using different kind of variety of data for PAG itself, but also different type of meanings assigned to the PAG-to-PAG relationship. And then we also did some additional work. I'll just briefly mention that this work is actually in press at the ACM IEEE Translations in Computational Biology. So we also asked the question, if there are R-type PAG-to-PAG relationship, then are there novel PAGs that's not already found? Especially, can you bundle together upstream genes all pointing to the same PAG, or downstream likewise? And then we call them virtual PAGs, something that nobody else has reported them, but through this relationship finding, we thought that it makes sense to bundle them together. So that paper essentially come up with an algorithm to find this virtual PAG to MPAG regulatory relationship by iteratively looking at all of these loose upstream packs and then see can we actually combine them together. As we combine them together, we want to preserve the cocoa or the biological cohesion relationship among genes inside these virtual packs. Okay, so I'll skip that, but I'll also show you that for the study that we've done, the virtual packs achieve really, those are the blue lines. When you look at the cocoa beans, they achieve a much better quality than the database of regular packs. So essentially, we are picking up significant information and we're putting them together, and nobody else has found them. So this is really not only a database issue, it also opened up new directions of research. In addition, we're also trying to find causal relationship, but I won't talk about it here. But if you want to actually look up the data, we have a database, essentially document the gene-to-gene pair that's more traditional. 84,000 packs of singleton that's essentially the gene themselves, or multiton that's regular packs. For the two type of relationship pair, we were able to extract 7 million significant M-type relationship and 120,000 R-type relationship. And what can we do with it that we don't know exactly, but I can show you one case study for it. Okay, so these are all the web interface. I think you can go online, you can search for this information. I won't show you the details, but essentially you can find the pack, and then you can use a visualization tool to look at connections. You can look at the matrix, how the enriched pack relate to each other through shared co-membership relationship. So in the figure that showed, so you can show intra-pack relationship, essentially a gene network inside a pack. Gene network, we also combine it with our ranking algorithm, essentially looking at the centrality of these genes and map them. So you not only see the connections, but also see the major connected hub. And they're all just done within one simple query. And then you can also look at the intra-pack relationship with your queries. And then once you zoom into a particular pack, you can look at all of the neighboring, either upstream or downstream regulators of the original query ones. So with this, we did a case study years ago. Essentially, we look at the myelin-derived suppressor cell microarray data, and we ask the question, what could we have done mapping to the conventional gene regulatory network? And then it found out that out of the 1,100 genes that are differentially expressed, we would have lost the majority of them and only kept a quarter of them in the network. But if we were to use this pack regulatory network, we would have retained most of them in the network analysis because of this integration of packs and network. This is a network that we generated. It still looks messy, but then this is all the data. Given this is a very comprehensive gene list with 1,000 genes, and we were able to reduce it to a diagram like this, where sitting in the center is a pack responsible for immunity functions, and it's right on target. We can see what are the upstream and downstream regulators. Remember I told you that we can also bundle all of these loose genes into virtual packs. So in the next version, we just converted from this to this with a lot more abbreviation. So I think with that, and I want to conclude this part of the analysis essentially to say that we're able to use this PAC and the PAC networks to gain, maybe help ourselves gain a better understanding of the global perspective. And this global perspective, you may say, well, this still doesn't help you answer the question what genes to use for precision medicine. But I would argue that when you're making decisions, you're making much better calculated decisions with minimal bias. Right? So it's like when you're trying to invest in the stock market, you can just pick a stock and then win as is you're winning a lottery, or you can actually systematically scan all of the sectors based on the current economy and then make a very informed, calculated bet on which sector of stock to invest. I think it's so I like to encourage people to give me feedback and then try out tools like this. But then I think that's still not quite satisfactory to us because these figures are still harder to understand and interpret, and it's not completely visual. And so I want to talk about the related branch of research, the terrain visual analytics. And visual analytics is, I think, is one of my favorite tools as a data scientist, because as a data scientist, I'm very familiar with working on this side. I think Ben just did a wonderful talk in the morning, NCBI's tool that can just marshal data through all kinds of pipelines and get to this point. But would you actually immediately use it to validate in the biology or clinical setting? And probably not, right? Because the biologists are not working still today in this data science mode. They're working in the mode of telling stories. Right? So if you give me some genes, and then I'm going to test it in the biochemistry lab, I'll do genetic knockout studies, I'm going to actually put it into just preclinical models, and then I can start, if I can start telling stories, and then I will try to fill in the gap. So visual analytics is a way to engage both, to essentially make use of machines and the human together to explore solution together. So this is one example. So if I don't tell you how I make this visual patterns possible, and you can probably still gain some ideas. I can tell you that these are 3 conditions of Alzheimer's disease at a molecular level. One is a mild cognitive impairment versus normal control. And this is a moderate Alzheimer's disease versus control. This is severe. When you're looking at these patterns and you say, oh, A1 and A2 peaks, they consistently show up across the spectrum. So they must be characteristic or potentially diagnostic. And then these peaks get replaced by later stage. So they may be used for staging of prognosis. And this peak actually increase in height. So I think immediately our humans, before deep learning, our humans are the best in perceiving these patterns. But I would still say that the human has a place in finding biological knowledge. And then the question is this really scientifically sound, or is it just a fake data? So it's not fake. Where's the data coming from? So we use protein interaction information. We use these all of this PAC information, and then we couple with functional genomic data or genomics data, and then we make it all together. So the basic representation of the figure, the panel that I showed you earlier, start with a network where the nodes our genes and the edges are functional PPIs. And the node size are all drawn according to their centrality measures. And I won't bother you with how we derive that. But essentially, we're assigning significant measure to nodes. And then you can actually map your omics data as different representations of the nodes and the node weight, edge, and the edge weight. So you can do it for the genetic mutation data or the functional genomic data. And then also underlying what we're trying to optimize is the layout. So it's, in order for pattern to show up, you can imagine that which genes are close to each other will pretty much determine whether there's a large peak, small peak, or valley, so on. So if that was not done carefully, then I think you will get a bad result. And we tried out the cytoscape, the organic force directed layout, and they were okay, but they were not ideal because they were never meant to do this kind of analysis where the information are encoded in the network nodes or edge weight. So we had to come up with our own. This is unpublished yet, but we got all the research done. Essentially, we use a energy field optimization method, and we encode all of the biological parameters, and we want to balance the hub-centered community, like here, versus space filling. So you can control the parameters and then get to either the things to center around the hub or just have very nice space filling features. And with that, we also allow us to incorporate the prior knowledge of gene set annotations. For example, you do a gene set enrichment analysis from pager, then you got these three. If you're just not optimizing them, then they're all over the places. But our layout allows you to also optimize based on the constraints of these communities, so you get more or less them together. So all of these series of optimizations are meant to help us improve the chance of discovering patterns from the data based on prior knowledge, right? So you can say the prior knowledge is all fake, but then I don't know what else to trust. So I think we're doing our best to incorporate everything. So if you start with a raw network, and then if you lay out optimally, rank the nodes, rank the edges in some cases, and then you get a pretty decent layout. Okay, and then I think one other things I would like to mention is how do we actually determine this pattern? So this pattern, if you have this A-gene network, where all of these are nicely laid out already, but they don't automatically come up with a pattern, right? So you have to actually tune in certain parameters so that they're more reaching out to each other to generate the group pattern. So in a sense, if you're tuning out this parameter, you're more likely doing a gene set based analysis. If you're on this side, then they're more like a single gene based analysis. How many more minutes do I have left? 10 or?
10.
Okay. All right. I can show you some case studies where we would apply this. One is we apply this to analysis of doxorubicin treatment on yeast cell growth. So this is just from a collaborator who is curious about what gene terrain can do. So we have two treatment versus control and glycolysis and versus respiration conditions. From his experiment, he would generate thousands of genes and lots of gene ontology categories. And normally you would get stuck here and not know what to do next. So we plot the gene terrains, and we found out that there are actually more or less features within the gene terrain. So we can actually just zoom into specific features and ignore the rest of the noisy peripheral regions. This is our way of cherry picking, but our way of cherry picking, as I said earlier, is best because this is a more informed decision. And when you look at the red lines of the gene ontology enrichment result versus the blue lines, the red lines without the gene terrain analysis, you're looking at everything. And with terrain analysis, you're carving out only the features. So you'll see that these gene ontology enrichment results are different now because you're zooming in on the features that are globally more suggestive of this biological condition. In fact, this pattern completely disappeared after the terrain analysis. Okay. Another example is we're trying to reanalyze the GWAS data, and there's a Nature paper 4 years ago that looked at all the GWAS study from multi-stage schizophrenia study with 36,000 cases and 113,000 controls. GWAS, you know, is always very messy, so people have to use very rigorous statistical measures. In the paper, they set the threshold at 5 times 10 to the negative 8, and then you get 128 SNPs, and then they did the assessment. So the Nature paper is never meant to be thorough, but it's meant to be just suggesting that there's a gold mine there, as we always know. And so what we did is that we significantly lowered the threshold to negative 4 and allow us a lot more number of SNPs to show up. But then we use a prior knowledge network to sift the genes that come down to about a little less than 1,000. And then we build a gene terrain, and then we look at these features. Right? These features, they're all GWAS SNP data aggregated around the gene regions. Right? So we're just counting the number of SNPs that pass through the threshold. And then when we do that, we were able to reconstruct almost all the important therapeutic target with very high RP score, which indicate the significance of these genes, and all the major categories, and plus a lot more, plus the relationship. Okay, here's another example. I think I can have the mouse. This example shows using gene terrain as a platform to do even some of the simulations. This is a simulation done on a molecular signaling network that we constructed for ER-positive breast cancer. Overall, to begin with, there's an overexpression of the ER signaling network, so its overexpression is red color. As you start the simulation, you see that the terrain shows things started to change, and this simulation is a tamoxifen treatment of the ER-positive breast cancer. So it shows it works, and it works because the network changed and the signal cascade through the network brought down the expression value. So I think that potentially could be used as a simulation tools, engineering, and biomedical engineering. There are lots of tools, but most of them are built at the cellular level and above. So I think this is our attempt to build something, even not at the differential equation level, but it's still it's helpful. And then the last example I want to show is it can also be used as a disease classification tool. So in this particular study, we look at the GEO database of non-alcoholic fatty liver disease samples. And then in that database, and they have liver biopsies of normal, obese, liver fibrosis, and the NASH patients. So we can actually extract these clusters and then build PPI network, and then from these we build gene terrain. In fact, we look at 32 samples of NASH and normal, and we build 32 gene terrain. And there are only two types that we use here. One is NASH, and the other is normal. So as it turned out, that you can see the pattern. Most of the normal follow this pattern, and most of the NASH follow a different pattern. So if I'm posing this, can deep learning do learning from 32 samples and achieve 100% sensitivity and specificity? I think that's impossible. But I'm not saying this can be generalizable to all problems domain, but I think at least this is very promising, because in order to generate these, we're using all of the human knowledge as a guidance to help us interpret these patterns. It's like a driving, and you have all the database of what the objects look like, and then that will make autopilot so easy. But if you were asked Tesla to learn the object as it starts driving, then you're going to crash so many times and have to run 1 million miles before it can achieve decent performance. So some final thoughts. Getting big data is easy, in my opinion, but reducing them is really hard because of the biases. So temporarily, we should be able to build descriptive models with inferencing capability. In the future, obviously, we want to build quantitative models. And then use both human intelligence and machine intelligence. That's my experience. With that, I want to thank all of these people that helped with this work and take some questions. Thank you. Yeah.
With respect to using the structure for classification, I suppose you probably recognize it yourself, because you said that the 100% sensitivity and specificity is definitely suspect. So I was wondering if you made any attempt to assess model overfit with respect to that?
Yeah, so I think what we did was, of course, this is a uniform data set. And when we learned the model, we have only 32 samples. So we just use five-fold cross-validation and then get this. This particular case, NASH and the normal, is probably easier to distinguish. You can see people with liver problems, and then lots of the genes will mess up. So it's a proof of concept, but the more tricky examples of what we're trying to do is to look at the glioblastoma, where if you go to TCGA, you will see that there's no survival markers. And so we're actually trying to use technology like this and see how much improvement. We do see improvement. It's not 100%. It's far from that, but I think we do see hopes in there. I hope I answered your question.
You did, thank you.
Yeah, great talk, thank you. I think there's the place for visual analytics right between, like you're describing. I think we're missing tools like that. I was wondering what's your opinion, because you didn't touch upon how important it is to have visual analytics tools that are interactive, to be able to select, to have a lasso, to filter, and that's one question. The second one is, what's your opinion on the 2D, 3D, whether we really need 3D displays or is two-dimensional sufficient? And
yeah, that's a very good question. The first one, Actually, as a disclaimer, I'm using the software that's I generated from my startup company, and then they're actually building this as an interactive platform. You can actually select data points, and you can query it and build terrain on top of one. But the one that I'm using is a free version. So the second question, what was the second question?
2D or 3D?
Oh, 2D or 3D, yes. Initially, we thought 3D was very important, but 3D, you can probably overlay some additional features, like you can use height and one feature, and then just color intensity as another feature. But there's only too much you can add between 2D and 3D. So instead of doing that, we thought that if you can actually build terrain of variability on one 2D and then the terrain of expression value on another, and then compare them, I think that's almost as good. So 3D or 2D, it's just a personal preference, but it's not a deal breaker.
Yeah, for me, 3D is difficult because of the interactivity issue. Selection becomes an issue of 6 dimensions, right? 6-dimensional device you need to select data,
Right.
so, yeah.
Yes.
So speaking in the precision medicine, I think there are also missing information here. Epigenetics is a big one, all kind of, another type of layers, metabolomic layers, another, post transcription or translation or modification is another. So many things are still need to be integrated. So what is your grand plan on that?
Yes.
And more related specific question is, if you want to pursue this network-based approach, added features may, I think, probably computation is less efficient for scalable because your network is just bigger and bigger and complex.
Okay.
So
Yeah, so
what was your strategy
good,
to deal
good.
with potential scalable computation?
Right. So the first is a multi-omics. We do have a strategy for different omics data set. For example, we're right now working on a microbiome just terrain, whereas we define each species identified as a node and the phylogenetic distance between species as edges. And then if you have quantitative information, then you can display them out here. And then if you want to build a community, then there's a lot of research on finding species that always co-express together, but then there's a human microbiome project that's generating a lot of data. So in a sense, I felt that this technology is, it can adapt to different omics, but not without additional research. Second is about scalability. I think that right now, if you think about the gene space, 20,000 genes, we were able to plot the terrain with the DEMA layout with just one single thread computer for 2,000 node network for around 20 seconds. And in fact, I'm going to collaborate with someone that you guys have a lot of expertise on HPC and graphic card. And I think we can bring down significant orders of magnitude faster or two. But in reality, when you measure all of these omics data that only measure one aspect of biology, I rarely see cases where you have to do whole omics scale network. I think a lot of the analysis you see out there, it looks nice on Science and Nature paper to have the whole map, but then they're less useful for specific analysis. So I think we're manageable with the technology improvement.
Thank you, Dr. Chen.